The second part of Unit 4 of the AP Bio curriculum is the cell cycle, which is in the same unit as cellular communications. So, this video we're going to be talking about cell cycle. So, cell cycle is important because cells they can't just live forever. Cells would need to divide. Cells divide every once in a while, but the dividing part is only a small part of the life cycle of a cell. Most of the time, like most of the lifetime of a cell, the cell is busy carrying out whatever process is intended to. And then there are also cells that are non-dividing, which example of that is red blood cells. They do not divide at all, and there are other cells that temporarily do not divide, and they enter a phase called G0. And yeah, we're gonna talk more about that later. So there are different phases of a cell. So the phases are interphase, and then so this is how it's usually represented, like in a circle, all right. And the cell is divided into like kind of two periods, and then inside the so then this is the big part. It's interphase, and then within that like small part there are two phases. So there is mitosis and then cytokinesis, and we're going to talk more about that in a second. So first, let's talk about interphase, which is when interphase is also called the resting phase. The resting phase of the cell, but it's not really the cell's not really resting. It's doing a lot of things. So the interphase is the longest phase of the cell cycle. During interphase, the cell grows so that it has enough material to divide between the two daughter cells. So like make sure it has enough uh enough genetic information, enough organelles to form two daughter cells. So when cells split, so one cell, when it becomes two, these are called daughter cells. And these are also daughter cells. So there are three phases for interphase. So interphase, it comes with, phases itself. There's the G1 phase, there's the S phase, and the G2 phase. So G stands for growth, and S stands for synthesis. So during the G1 phase, the cell grows and prepares for the replication of DNA, and then there are some cellular organelles that are replicated during the G1 phase. And then S, synthesis, is when it's a very important part of the phase, which is when the cell replicates its genetic material, it duplicates all of its chromosomes, and during the interphase, during like the S phase, every single chromosome is duplicated so that a copy can be used, so that there are two copies. One can be used for one daughter cell, and then one can be used for another daughter cell. So these are like chromatids, right? This is how it's usually like represented. So it's kind of like this, and then there is a centromere. This is a single chromatid. This by itself is also called a chromosome. And then when it replicates, you're gonna learn more about in a future unit. I forgot, it was like unit 6 or something, where you're gonna replicate the DNA replication, and then when it replicates, it kind of becomes two. So I'm gonna use a different color, but the red and the blue, they are the same. All right, these are the same, and then the center here is called the centromere. And then these two, they're also called a chromosome. All right, but this over here, so this and this has the same exact like genetic information. So that's when like DNA is replicated, and then it's still one chromosome, and then later during mitosis it's gonna be separated. So These duplicates, they are still linked. They are basically like conjoined twins. When you divide it, these are called sister chromatids. Chromatids. All right, and they're held together by a centromere. And during the cell cycle, there are regulations. There are proteins that regulate it, make sure that damaged DNA gets fixed, or that like so there are different proteins that is used. There's like P53. There's the RAS protein where it regulates like whether the cell can move on to the next part of the cell cycle or not. The protein might trigger a response that fixes the DNA or whatever. Because when you replicate a cell, you don't want messed up DNA to be produced in one of your daughter cells. So then that is important. And then there's cells that look that like see the cell. Oh, it's too damaged. It can't be fixed any further. It will trigger a response called apoptosis, which is programmed cell death. So those are the cell cycle checkpoints that make sure that cell division is happening properly in eukaryotic cells, and that if there's a problem in the progression through the cell cycle, it will stop immediately. It will try to fix the issue, or if it can't be fixed, it will just tell the cell to kill itself. Yeah, I think that is it for interface. So, cancer is where a cell has a kind of like a mutation in the genes where it's like so there can be like a mutation in the protein that controls the progression through the cell cycle. It might result in unregulated cell division. That is cancer. Cancer is it happens when like normal cells they start behaving and growing abnormally and spread to other parts of the cell, that other parts of the body. That is cancer. And then it's caused by like mutated genes that induce cancer, and those genes are called oncogenes. And onco is a prefix for cancer. So oncology, oncologist, you go see an oncologist if you have cancer. And those genes, no, normally before those genes become oncogenes, they are required for proper growth of a cell and regulation for cell cycle. But then during like replication or something, the genes are mutated. They become oncogenes because the genes they are no longer normal. They are different from normal, and they convert normal cells into cancerous cells because these cells they no longer function normally. They are abnormal, and yeah. The normal version of the gene is called proto-oncogene. So proto-oncogene, they are the healthy ones, healthy slash normal ones, whereas oncogene is just the cancerous ones. Cancer. Alright, and the different parts that you check over it and blah blah blah. And now the most interesting part, which is my favorite part, is mitosis. Mitosis. Mitosis has different phases. An acronym that I remember it is PMAT. So those are the four phases of mitosis, and in order. So P stands for prophase, M stands for metaphase, A stands for anaphase, and then T stands for telophase. So prophase is when the nuclear envelope kind of dissolves. So let's say we have a cell here, all right, and this is the nucleus. So we're gonna make it have 4 DNAs. Those are the 4 colors I'm gonna use. So it has like this. So DNA, they're like all over the place because it has to be like wind up so that it can be easily read. So these are like the four like chromatins that we have. So this is prophase. The basically like the nuclear envelope dissolves, and then now whoop, it's completely gone. So then that's prophase. It's when the nuclear envelope is dissolved or it disappears. So the nucleus is broken open. All right, and then now, well, it's also important that they become like coiled first. So they have to be coiled up into like condensed chromosomes. So let me actually change that so it looks more like this, which is better for replication. So if you remember, they look like X shapes. They look X shaped. So these are like chromosomes, all right, with like sister chromatids. They're all different sizes. Some can be big, some can be small. They're like X shaped. They're actually more like this, but oh well. I'm not an artist. So then now, during metaphase, they line up in the middle. They line up in the metaphase plate. That is the fancy word for like the equator of the cell. So it line up in the middle. Lined up in the middle. So middle. That's how you can remember it. That's how I remembered it. So metaphase. And then it's connected. And then now there are these spindle fibers that connect to ends of the chromosomes so that it can be pulled apart. And this is the next phase, which is anaphase, which the chromosomes are pulled apart. So it's gonna look something like this. I'm gonna make this a little bit wider. So it's gonna be like split up like that. All right, like that. It's being pulled apart, so that's why it looks like this. And they're all like different shapes. And it's being pulled apart and pulled apart. So anaphase means apart. Prophase, you can maybe remember as like prep, right? And then metaphase is middle. And then anaphase is apart. And that's when the chromosomes are pulled apart. And now these are chromosomes on its own. So the sister chromatids, they are chromosomes on its own. They each have a centromere, sorry. Right? They each have one. So they can be a chromosome on its own. And now the last phase is just telophase, where the nuclear envelope forms between the two cells. So during this process, these cells they're still one cell. They're still one cell. They're still one cell. But then now they're like apart now, so they're just they're apart now. All right, they're a little bit messy. They're gonna start like unwinding up so the cells can read it. But then now during the telophase, a nuclear envelope forms. So I'm going to use a nuclear envelope forms. And now after mitosis, cytokinesis happens where it's being pinched down the middle and then it forms two cells. And if you remember, these are daughter cells. And it has a nucleus with a complete set of uh, genetic information. And then these are like unwinded up so that it can be read by the cell for protein production. And that is the process of mitosis. In animal cells, there is something called cleavage furrow that happens, but then in plant cells, they have a cell wall, so that can't really happen. So a cell plate is formed down the middle region, like over here, where it just like splits off, and yeah, there are two cells. So the purpose of mitosis is to produce two daughter cells that are identical to each other, and yeah. Mitosis happens in every cell, like practically every cell, except for sex cells, which are like sperm and eggs. It goes through a process of meiosis, which is slightly different. It's different than uh, mitosis. And that's it for this video.